Hello, my name is Lee Torres. I am a professor at Oregon State University and I work within the Marine Mammal Institute there, and I lead the GEM Lab. I'm really excited to be here today to give the keynote address at the 2020 State of the Coast Conference. Yeah, I feel quite honored to be here. It's one of my favorite conferences, honestly. I remember attending this conference when I first moved to Oregon in 2014, and it was just such an amazing opportunity for me to learn so much about the science and management and stakeholders of the Oregon coast. It's a really beautiful integration of all those elements. It brings people together from all sorts of backgrounds and interests in a great way to really share information and communicate. So yeah, I hope this conference is just as fulfilling and exciting to everybody. I know it's difficult times and having it remote is odd, but yeah, I'm sure we will all learn and share and gain from it as well. So thanks to the organizers for inviting me, Oregon Sea Grant. I'm sure it'll be another great conference. So yeah, since I arrived here in Oregon, I've been leading a number of different research projects centering around the gray whales that feed in our coastal waters here in Oregon during the spring, summer, fall months. So that's what I'm going to be talking a bit about today. But first I want to thank all the co-authors that have helped me conduct all the research, which are listed here. In particular, I want to call out the students, Clara Bird, Alejandro Fernandez Ajo, Lisa Hildebrand, Leila Lemos, and Florence Sullivan. They've all been really integral in the research that I will be presenting today, so I'm grateful for their collaboration. All right, so let's jump into it by starting with who are the Oregon gray whales? Well, the whales that spend their time here during these summer months feeding are called the Pacific Coast Feeding Group. And they're really a subpopulation of the larger eastern North Pacific population of gray whales that migrate from Baja California here up to the Alaskan Arctic to feed. But this subpopulation is about 200 to 300 animals that instead of continuing the full migration up, they feed in this region right here between sort of British Columbia and Northern California. So the whales show high site fidelity to this region, meaning that they're seen year after year after year feeding in this area. Although they do move between areas, they don't just stay in one spot, but they have been seen year after year within that area. And they're thought to feed primarily on mysids, which are little shrimp-like zooplankton. So, but what we don't know about this group is actually quite a lot. We don't know. We don't really know what their connection is to that bigger group of gray whales, that eastern North Pacific population, or how much mixing there is reproductively. We don't really know about how the population is growing in terms of recruitment of calves or immigration of other adults to the population. We'd like to know more about their foraging preferences and their ecology and behavior, as well as their distribution throughout this area and their habitat use patterns. And also we need to know more about the anthropogenic impacts on their health and well-being. So the work that I've been doing here mainly centers around this question about describing gray whale physiological response to stressors. And so this is really lacking for all baleen whales. And what we focus on for our work is, the main point is trying to figure out what the role of increasing ocean noise is on whale stress. And this is important because ocean noise has been increasing quite significantly in the oceans for the past few decades. And while there have been some studies that have shown behavioral response of baleen whales to increasing ocean noise, there really has only been very few, one in particular study that showed a link between their physiology, the stress of whales, and ocean noise variability. And that's because studying cetacean physiology is quite hard. You can't capture these large cetaceans and you can't take blood from them regularly. So it's quite hard to study them. And also physiology in general is a hard thing to study because there's lots of intrinsic and extrinsic variables that can affect an animal's health. So, you know, just their reproductive state, their age, how healthy they are. So if they have an injury, all of that can impact an animal's stress. So what we try to do in this study is concurrently examine the ecology, behavior, environment, life history of the animals as well as the animal's physiological stress. So we're trying to bring all of it together to understand what the multiple stressors, how multiple stressors are impacting whale stress. And we need this information because physiological response of cetaceans to noise is really needed. We need that information to inform these PCOD and PCOMS frameworks, which managers use. Now, most of you out there are probably saying, what is PCOD and PCOMS? And I don't blame you. So PCOD is the population consequences of disturbance, and PCOMS is the second iteration of that, which is the population consequences of multiple stressors. Now, many of you are probably still saying, what is that? Well, it comes down to this framework here, which thinks about all the different sort of stressors to which an individual could be exposed. So if you think about what's in this white box as the individual, an individual's physiology, behavior, health, and the vital rates can all be influenced by a stressor. But when managers manage populations, species for conservation and management purposes, they do it at a population level. They don't manage individuals. So what these PCOD and PCOMS frameworks do are trying to understand how individual response to stressors then end up affecting population dynamics. So whether a population is increasing in abundance or decreasing or staying the same. So this is what we're trying to do here, is fill in some of these gaps about these individual response so that we can understand how the population is responding to different stressors that they might be facing. So we're using these Oregon gray whales as our study system. And this is because of the reason I'll show you here, not necessarily because they're the most stressed out population of baleen whales, but mostly because we have good history of these animals. So many of these animals that we study have been studied for 30 years. So because of that, we have a lot of information on individual sex and age. We also can collect replicate samples from whales, individual whales, because we know we can see them year after year and month after month in these coastal regions. So this, I should show, is our sort of study area, which goes from just north of Depot Bay down to past Seal Rock. And it's about a 45-kilometer area, and it's very coastal. So these animals are very coastally oriented in their foraging. And also because this region has variable soundscape. So we are again interested in the impact of variable noise on their physiology. So this region isn't by any means the loudest part of the ocean that we would encounter, but it does have lots of variability, which means some quiet periods as well as some loud periods due to fisheries openers or commercial, shipping or construction. So, and of course, wind and storm events impact that. So we're really interested in how that variability impacts whale physiology. So this is the framework that we have set up for ourselves to try and understand whale physiology. So at the center of it is stress. We're looking at the stress response of whales to multiple stressors. And some of these stressors can come from the soundscape, so anthropogenic activities such as vessel traffic, marine construction, as well as natural forces like wind, and then predators like killer whale sounds can certainly stress out these whales. We're also interested in how individual demography, so the age and sex and reproductive state of the animal, might influence their stress levels. And then the overall health of the individual can certainly impact an individual's physiological state. So whether they're suffering from an injury or poor nutrition, or what their body condition is like. Clearly, the oceanography that impacts their prey availability can certainly impact their physiology. So we are monitoring that as well. The behavior state of the individual is another aspect that we monitor. And then, of course, all the temporal aspects are included in all of this work that we do. So because we're assessing all these multiple stressors, we're also using multiple tools to try to do this. So in the center of it, we're using fecal hormones, so in particular cortisol. And we're using this fecal hormones because it's a non-invasive biological material that we can collect repeatedly from individual whales. And also because it's been shown in a number of studies from terrestrial animals that the cortisol levels in fecal samples from individuals is a really good indicator of their stress response to various impacts. And as we know from a lot of research on humans in the biomedical field, is that when we're stressed out, we tend to be more susceptible to disease and viruses and have poor we can have heart disease and so forth. So certainly if we have chronic stress, that can impact our health. And so we're using cortisol as the response metric to try and understand how stressed out whales might be. So we have hydrophones in our study area to understand the soundscape. We use a lot of photo ID and life history to understand the demography of the individuals, as well as their reproductive hormones that we get also from the fecal samples. We're using photogrammetry that we collect from a drone to look at their body condition, and again, photo analysis to look at if the individual has an injury. And also the fecal hormone thyroid, T3, is an indicator, we think, possibly of nutritional state. So we're also using that to look at the individual whale health. For oceanography, we're using a lot of sensor data, as well as prey sampling. We again are using drone observation, so unoccupied aerial systems, that's UAS, and as well as field observations to monitor the behavior state of the individuals. So we've been doing this for 5 years now, since 2016, and which has allowed us to really look at how individuals change over time, because within that time period, individuals can move between reproductive stages. They certainly change age, as well as their nutritional state can change. So just as an example of our ability to collect that replicate information from individuals, this is a table of some of our whales that we've been studying. So this is their name over here on the left. And then the number of fecal samples we've collected from individuals over our 5 years of sampling. And so what you can see here is that this individual, Soleil, we've collected 19 fecal samples from her over the 5 years, which really allows us to dig pretty deep into how this individual's hormones might have changed naturally over time, as well as to potential unnatural influences. And then we have some other individuals which we've also been able to collect at least one fecal sample in each year of our study. So again, this is really valuable information that's helping us piece together the story of whale physiology. So if we circle back to this framework that I just walked through, it's quite complicated. And then the idea is to integrate all of these different aspects to understand how multiple stressors might be influencing whale physiology. But what we first need to do is understand the sort of unidisciplinary results. We first have to understand what the baseline levels in body condition and hormones and so forth are in order for us to understand what might be change or what might be an abnormality. So to start here, I'm going to walk through some of these unidisciplinary results that we've uncovered in the last couple of years. So we're going to start with some of our hormone results coming from the fecal analysis. So this little video clip shows you how we collect the fecal samples. It's quite opportunistic. We wait to see the whale poop, and then we zoom over on our zippy little rib and collect the fecal sample with our nets. And so you can see there a fecal sample in this image on the right. And we're looking at 4 primary hormones within these fecal samples. So cortisol, which is the stress hormone, progesterone and testosterone, which are the reproductive hormones. And then we're using this thyroid hormone T3 as potentially an indicator of the nutritional stress of state of the whale. And it's important to know that all of this work is really brand new, particularly for gray whales, but for almost all whales, using fecal hormones to understand their hormone variation is quite new. So as I walk through these results, keep in mind that this is really quite novel information. So here are some of those results. So on the left, what you see is a nice positive relationship between the hormones progesterone and testosterone, so they're reproductive hormones. And this is to be expected. This is quite normal in mammals, is to see a positive relationship that as testosterone increases, so does progesterone. But what's interesting here is to see that the relationship between these thyroid hormones and cortisol. While this relationship isn't quite significant or as significant as this relationship on the left, what you do see is a positive trend in both years that we assessed these hormones, that as cortisol increases, so does this hormone T3. So again, we're just starting to understand what sort of the natural variation in their hormone levels are like. So here's another way of looking at this data. What we did here was look at how progesterone and testosterone, so the two reproductive hormones, vary by demographic group. So we have immature males, an injured male here that I'll talk about a little bit more later, immature females, mature males, resting females, pregnant females, lactating females, and post-weaning females. So what you can see here is, particularly here, the high progesterone levels really belong to these pregnant and post-weaning groups, which is what we'd expect, and it's good to see. And then this one is also quite interesting. The really high testosterone levels are all within this mature male group, and really quite high levels sometimes. And that indicates that those males are in a reproductive state and trying to mate with females. So this is another way to look at the data that we've collected. So each one of these colored humps here is a year, the distribution of cortisol collected within our 3 different years. Samples collected within those years. And so overall, this black line represents the mean of all those values. And then this is the second standard deviation. The red line is the first standard deviation. The gray line is the second standard deviation above the mean. So what we're trying to do here is understand exactly what is stress, like what is a normal stress level for these animals and what might be considered a high stress level. So looking at this distribution plots, we might think, okay, well, if this is the average stress level, something over here probably indicates that the whale is stressed out. And what we were able to do with a really unique situation with this whale Equal is try to validate our distribution, our understanding of the distribution of stress levels for these whales. So we saw Equal on the 19th of June. It looked very much like a happy normal whale, but then we saw the whale again the next day, and we noticed that it had some fresh scars on it, which we think is likely from a vessel strike. And we were able to collect a fecal sample from Equal on this day. So we know that this animal endured a significant injury within the 24-hour period preceding that fecal sample collection. And if we plot where that the cortisol value for that animal lies, you can see that it's definitely above the average value and close to this first standard deviation. So yeah, we're beginning to understand what a stressed out cortisol value is for gray whales. And then we also have been able to collect or analyze fecal samples from some stranded individuals. So 4 individuals, 3 of which again are all around, their cortisol values are all around this this first standard deviation line. This one we think might have had adrenal failure or something, because clearly it's on the low end. But again, all this data is really helping us understand what baseline values are of these hormones, as well as what the high end of stress might actually be for these whales. Okay, so now we're going to switch gears and talk a little bit about the soundscape and some of our results from that. So we've had 2 hydrophones in the water each year of our study, one up by the Otter Rock Marine Reserve, which we are sort of our quieter area, and then another one near the entrance to the Newport Jetty here, which is more of our loud area because there's a lot of boat traffic in and out of the harbor. And you can see that these 2 hydrophones are only 17 kilometers apart, but also span the area of where we've managed to collect our fecal samples. So here are some of the results. And what I want you to understand, these 2 plots, these colorful bluish plots, are called spectrograms. And they both have the date on the x-axis here, and then frequency of the noise band on the y-axis. And then the coloration of both plots indicates the decibel, so the strength of the noise within that frequency band. So the bluer the colors, the quieter it is. And then the brighter colors means it's quite loud in that area. So across the time span, so this is the Newport hydrophone, and the top one is the one near the Marine Reserve. You can see that these bright blue stripes here sort of happen at the same time at both hydrophones, and that's indicative of wind events, where it's sort of an increased noise across all the frequency bands, and those happen at the same time. But what you can also see is these bright yellow and green colors in the Newport hydrophone recordings. And those are indicative of louder noise events in the lower frequency bands caused by increased vessel traffic across that area. So another way to look at it is through these two plots, which the top one here shows that as wind speed increases, the noise increases. So that's what you see here. When the wind speed is greater than 4 meters per second, this black line, you can see that the noise increases. And what this plot down here shows is that we were able to decompose the noise signal that we recorded at the Newport hydrophone. And the blue line indicates the variability over time from wind. So you can see on these days here, the wind level was quite low, but what you see is the rest of the noise was quite high and variable. And we think that the rest of the noise is basically from increased or pulsed events in vessel traffic going by on these days. But what's interesting is that you can see here that when wind noise increases, so we had a wind event or a storm event on these days, you can see that that residual noise decreases, and that's probably because there were less boats on the water because it was windy conditions. So anyway, these information is really helping us understand how variable the soundscape is and what might be contributing to the whale's response to sound in their environment. Because these animals, it's important to remember that they're incredibly acoustically sensitive and active. They use sound to forage, to communicate, to mate, and potentially to navigate. So really when there's a lot of noise, added noise in their environment from other factors, it could really impact their natural lives and their ability to feed and reproduce and communicate. So we also did this daily relationship. We looked at the relationship between the noise that we're recording on our hydrophones and the number of vessels on the water. So the Oregon Department of Fish and Wildlife, they count the number of boats that leave the Newport Harbor and the Depot Bay Harbor every day. And what we did, we just ran these simple linear relationships between those counts of vessels and our acoustic recording. And sure enough, there was a very highly significant relationship between the number of boats recorded leaving the harbor and the noise that we recorded on our hydrophones. Okay, so now we're gonna switch gears and talk about whale body condition a little bit. So we measure their body condition to know how fat or skinny they are using drones. And here we have a flight where you can see we're heading over to this whale in the middle of the video here that's about to surface. You can see the jetty there with the dredge operating, which is adding noise to the environment. And so when we get over the whale, what we want to do is get it flat and straight at the surface, and then we take a still image of the whale in this sort of flat position, and then we bring this image into a custom-made computer program that we've developed where we measure the length of the whale as well as its body curvature between the 20% and 60% length of its body. And we've developed this metric called the body area index, which is similar to the body mass index in humans in the sense that it's all scaled relative to the length of the whale. So you can see here that we're dividing it by the whale length. And that's like in the body mass index, everything is scaled by the height of humans. And what this allows us to do is then compare the body condition, so the body area index of whales that are different lengths. And so it's all standardized. And so then we can compare across the same individual over time, as well as individuals of different lengths and demographic stage. So what you can see here is that this whale on the left here is sort of a skinny whale compared to this fat whale on the right. And you can see that the body area index metric is quite different. It's much lower for that skinny whale. So we use this BAI as a measurement of their body condition that we can compare between whales. And here you can see some of those results. So we have body area index on the left here on the y-axis, and again, demographic unit on the x-axis. And what we see is that the calves are quite fat and happy, which is what we'd expect. We also see that our pregnant females are much fatter than the other individuals. The mature males and the resting females are sort of in the middle. And then the lactating females, which are those females that are giving a lot of their nutritional body mass to their calf, are the skinniest. And so we see this nice trend that we would expect. Now here's an example of looking at using BAI to look at individual change over time. So this is this individual clouds that we saw early on in June of 2016, and when it was lactating. So you can see that it's quite skinny, clouds is, and you can also see its calf just underneath her. But then later on in the season, in that same year in 2016, you can see that the whale has regained a lot of its body mass through foraging. And indeed, the body mass index has increased over this time period. Sorry, not the body mass, the body area index has increased from 32 to just about 40. Now, we also use the body area index to look at how body condition changes throughout a foraging season. And what you can see here is that throughout the different months of the year, from when they arrive in early June to about when they leave in October, over time, the whales do increase their body area index, their body condition increases. And this is what we expect to see because they're supposed to be gaining mass here while they're on their feeding grounds. But you can also see that there's some variation in that. Like this year of 2016, you can see that in general, they're pretty much in good body condition all year round compared to the other years. So this got us thinking, well, what might be influencing this variation in whale body condition? So we had a hunch that it was related potentially to ocean conditions that are driving their prey availability. And indeed, what we think is going on here are some carryover effects. So I'll try to explain this here. So this is the population level body area index in the 4 different years of our study. And you can see in 2016, they were significantly better body condition than in 2017. And what we think is going on here is that in the years preceding 2016, so 2013, 14, and 15, you can see that these the upwelling conditions were above the mean. So let me stop and let me pause here and explain this graph here on the right. So this is the cumulative upwelling index. So you can use this metric to sort of infer how much productivity is in our ecosystem here. So higher amount of upwelling, probably better productivity and better prey is our assumption and hypothesis that we're working on here. And then this is the day of the year. So you can see here that in those years of 2013 and 14, we had really high, and 15, we had very high upwelling conditions. So the black line is the mean of these years. And indeed, the whales in 2016 returned in better body condition. Now in 2016, the upwelling conditions were very poor. You can see how low those upwelling conditions were. So that means that they were probably not foraging well in 2016. And so when they showed up in 2017, after their migration down to Baja and back, they were in pretty poor body condition. And then in 2018 and 19, you can see that the upwelling conditions were poor again in 2017, but then regained to about normal in 2018. And we see that the whales sort of started to come back in their body condition and improve again to be more like their 2016 body conditions. So we're still trying to work out the mechanisms here, but this is one of our hypotheses that we're working with at the moment. So this leads me into talking a little bit more about our work looking at the fine scale foraging ecology of gray whales within Oregon. And a lot of this work is happening down in Port Orford. And we have 2 parts to this project. One is prey assessment, which we do from a research kayak out there. And then the other side of the project, and these 2 sides of the project happen simultaneously, is that we're tracking whale movements using a theodolite. So I'll explain all of this. So from the kayak, we collect zooplankton samples, and from those samples, we look at the quality of prey. So what type of prey is there, and what is the caloric value of those different species of prey. And then we also do GoPro camera drops from the kayak. And with these, we're looking at how much prey is in the water column. So the relative abundance of prey available to whales. And then we use this instrument called a theodolite to track whale movements and behavior from a cliffside location. So these are some of our methods to collect the prey. So here you can see we have our little zooplankton net and some zooplankton in it, as well as we use a light trap sometimes to collect prey. And then later on, we identify each one of the zooplankton that we have captured to species. And then my student, Lisa Hildebrand, has produced these really interesting preliminary results that compare the caloric value of some of the different zooplankton species that we've collected. And what you can see here is that there's quite a big difference in the caloric value by the species of zooplankton that we collect. So one of the questions we're trying to address is whether the whales have foraging preferences. Basically, are they eating food that is more calorically rich, or where there is more food, or both? So this is one of our GoPro drops that we do, and you can see that this is sort of the underwater habitat where the whales feed and live during the summer months off our Oregon coast. You can see it's in these kelpie habitats. All this stuff in the water, that's the zooplankton prey that the whales are going for. It's a dense aggregation there of mysids of some kind. You can also see some rockfish in there because the rockfish also love those mysids. And so what we do with those GoPro drops is we take still images in a standardized method, and then we overlay a grid like this over it. And within each one of these grid cells, we score it. It's a relative score of 0 to 5 about the density of zooplankton in each one of those grid cells. And then we sum that, or we take the mean of those grid cells. And we do that for each one of our drops across our study area in a very standardized way. And then we compare that relative abundance information from the GoPro information with the caloric value from the zooplankton toes in that area. And we're able to generate these grid layers of caloric abundance. So if you think about this as sort of a prayscape map of the amount of calories in the area that a whale might encounter on that day. And so this, what this is, this yellow star demarcates where we do our theodolite tracking of the whales. So this is the coastline, this black line is a coastline. These blue numbers are our zooplankton sampling station. So we have some sampling stations over here that you can't see because our grid is overlaid on it. But what you can see in the grid is that these green and yellow cells indicate that higher caloric abundance available there compared to this side of the study area. So that's the sort of prey side of the project down here, but this is the theodolite tracking side of the project, which you can see here, one of our students using the theodolite instrument. And what it is, you look through this scope here, and essentially you focus it on the whale in the water, and you take a position. And then based on the known altitude of that theodolite and the angle to the whale, you can triangulate the position of the whale. So through this, we connect a bunch of dots together like this to make the track line of the whale's movements. And so this is an example track that we recorded from this observation point here, where you can see that the whale moved in. So even you out there, I'm sure, can see that this is probably the whale just traveling along. But then when it gets to this area, it starts doing more interesting things, potentially searching or feeding. And so what we can do now is overlay these two data sets, the prey data set with the whale movement data set, to see how whales change their behavior when they encounter different preyscapes. So what we see here is that the whale moves into this area, and then as it comes to this area with more caloric abundance, relative caloric abundance, the whale starts searching and foraging. So from this data set, we're able to understand how whales are making decisions based on the prey that's available to them. All right, so here we're going to take a little breather and watch a whale breach. I just figured everybody out there needs a little bit of stunning beauty every now and then. I'll play it again. We just got lucky at this one time when we were flying the drone and happened to catch this whale breach out of the water. So I'll take a drink of water and you guys can watch this one more time. Every now and then you get lucky and catch something amazing like that. All right, carrying on. Okay, so we're going to go into some of our multidisciplinary results now. So one of our primary questions are skinny whales stressed out? And so the answer seems to be yes, which is very interesting. So you can see here that whales that have a lower body index have higher cortisol levels, fecal cortisol levels. And this is a very significant relationship across all years. So that is very interesting to us and tells us that it is important to continue to monitor body condition as well as their cortisol levels, because the two are very much correlated. So another one of our multidisciplinary questions, obviously, is does elevated noise stress out whales? And so what we used here for this first assessment is to use the number of vessel counts as a proxy for noise exposure, because we know that from some of those earlier results that I showed you that there's a nice tight relationship between noise levels and vessel counts. And what we found here with our modeling results is that the vessel count on the previous day and the sex of the individual whale are really the primary correlates to increasing cortisol levels, which is pretty interesting. You can see here that as the vessel counts on the previous day increase, the cortisol level increases. And we think this previous day is because it takes a while for gut transit time. You know, you eat something and then it gets mixed up with your blood and your hormones, and then when it comes out the other side, that process we think takes about 24 hours. So if noise is increased on one day, you might not, what we theorize is that we're likely not to detect that increased cortisol or that change in hormone level until we collect that fecal samples 24 hours later. Now, we did test a whole range of different time frames, and this 24-hour period lag really was the most significant by far. And then when we look at it by sex, this breakdown by sex, it's pretty interesting. What we see here is that it appears to be those males that are having the biggest increase in their cortisol levels relative to the vessel counts on the waters, and not the resting females, which is interesting. And we're not entirely sure why this result is popping up. And we think maybe it's due to males being more acoustically active to try and attract females. And if there's added noise in their environment, maybe they're not able to attract females as well, or they have to expend more energy to make their calls to attract, to be reproductively successful. Again, a hypothesis that we're working on. Okay, so I just want to circle back to this framework, because we've been talking a lot about physiological change and the health of the individuals, but what about the behavior? And so I want to touch on this just for a little bit, because this is another really important aspect of our work. And we're doing this with our drone observations, because what we've been able to show is that using unoccupied aerial systems that are much safer, affordable, and efficient than, say, helicopters, for aerial observations, what we find is that we are able to increase our visible observation of whale behavior by 3 times using drones, because we're able to look through the water column at their behavior. So I'm going to switch gears for just a second here and show you this video that is a compilation of the different behaviors we've been able to observe using the drones. So here you can see some nursing behavior of the calf going underneath its mother. Here is some interesting behavior between 2 sort of teenagers that are engaging in some promiscuous behavior. You can see one whale kind of rising up and putting its belly to belly to the whale on top there. So we do see some interesting social behaviors. Here's another really interesting one. We are following this large female around with the drone when out of nowhere this other whale underneath came in and sort of, you can see, just jams right into its body of the other older female. So probably trying to get its attention for one reason or another. So, and then here are some of the foraging behaviors that we've been able to document. So that's a lower jaw flex, where you can see the puff sort of that lower jaw. Here the whale is skimming the surface. There's some zooplankton at the surface that the whale is sort of skim feeding on. Here the whale is, when it comes up to the surface, we often see this, a string of sort of bubbles coming out of the mouth, followed by a stream of sediment. So we've been able to document all these different foraging behaviors and tactics with these drones, which is really helping us understand whale behavior and how they capture and find food. So here's an example of a whale coming up after benthic feeding. So you can see it's just dispelling a lot of that sediment out of its mouth. Here are some headstands. So these are 2 whales engaged in headstanding behavior in a kelp area. And so you can see their head down and tail up, trying to nose into some prey patches down below. So we see that behavior quite a bit, these headstands, we call them. Here's an individual swimming on its side and opening its mouth as it's cruising through the water. So you can see its mouth open. Here's another slow-mo there. It's every time it sort of curves its body, it sort of flexes its mouth open. This is a whale that we filmed for about 3.5 minutes, swimming upside down and feeding. So we're not entirely sure why it was upside down, but we have seen that a number of times. Obviously, we also filmed some defecation events, which help us collect the fecal samples, which is great. This bubble bath behavior occurs quite a bit in the kelp areas. We're not entirely sure how the whales use it, but they definitely use it a lot to capture prey. And then this really cool jaw snapping behavior where you can see the whales just sort of trying to capture some zooplankton in its mouth. So this is, yeah, just a few examples that hopefully give you a sense of how dynamic and bendy these whales are and how they're really using quite a variety of different methods to feed within our coastal environments. So just to wrap up here, let's see. Oh, okay, so just to give you a sense of what were some of the other tools, because we're really quite interested in how gray whales find and capture their prey. And so what you can see here in this little video clip is we got lucky once to film this gray whale scraping the bottom trying to feed. And so this gave us the idea to try and put down some stationary cameras over time on a duty cycle. And what this is one of our first success. So this is just a pilot effort, but we wanted to see if we could get lucky enough to film a whale actually feeding to help us understand how they capture zooplankton. So this isn't quite the money shot, as we call it, but what you can see here is some zooplankton, and here they start to freak out and move around. And here comes a whale right through the image. And then after a little bit, the zooplankton come back together. So this hopefully we're hoping to use these underwater cameras more and to help us understand how the whales are feeding and what the relationship is between kelp and their zooplankton prey. So more to come on this. Okay, so a bit of a summary about the knowledge that we've gained so far. Well, what we've seen is certainly that whale stress is related to a number of factors. So age, sex, the reproductive class, their body condition, whether they have an injury, how much vessel traffic there is. So there's a lot of things that we need to pay attention to, including the fact that prey quality and quantity vary across multiple scales. So they vary, you know, within a few meters of each other, as well as between months and weeks and years. And we think that the movement patterns of whales is certainly related to the availability of different species and calories and density. So we're trying to figure out what the variation and all that is, and how resilient whales are to that variation. And we know what we're learning is that the ocean conditions are certainly related to body condition through that mechanism, through prey availability. So overall, what we know now is that we have to account for all of these factors when we're trying to understand whale physiology related to noise. We can't just assume that any increase in stress is due to noise. There's many other factors that we need to account for. So through this replicate sampling, we're getting closer and closer to finding out what those many relationships are to help us understand how noise might impact the whales. So what are we missing? Where are we trying to go still? Well, how much stress is too much stress? So we all probably have some stress that we deal with daily, all the time, especially nowadays. And it's likely true for the whales. So having stress isn't necessarily a bad thing, but there's a limit, and we need to know what that limit is. And then also when it comes down to noise, like how much noise is too much noise? So is there certain frequencies or intensity or duration of the noise that are really bad? And this information can be really helpful for managers to try and limit and regulate noise in very noisy environments. And of course, we're still trying to figure out what the relationship is between all these various behavior patterns, space use, the habitat, and the prey in those areas. And where we're trying to get to more and more is trying to understand how various impacts might be influencing the population dynamics of these PCFG whales. You know, does changing ocean conditions and prey availability, vessel traffic, noise, injuries, and entanglements, how are those affecting the survival rates and the calving rates of all these whales? So hopefully in the next 4 or 5 years, we'll be able to tackle some of these questions and get some better answers for you. And if you invite me back then, I'll give you some more answers, hopefully. So with that, I'll wrap up by thanking so many people and organizations that have really supported this work over time. There are too many people really to list, but I am incredibly grateful for the collaboration and the support. And again, I want to thank Oregon Sea Grant for inviting me here to tell you about all this work. So with that, I'll just say thank you very much for your time and paying attention.